Very good morning. It's really early morning here. So thanks to the organizer for giving me an opportunity to talk about my work. So this is my postdoctoral work that I'm currently pursuing, and today I'm going to talk about like how co-condensation of protein and DNA can lead to gene protection and shape of the nucleus. Before I move forward, I want to emphasize, like I want to spend one slide. What do you mean? What do I mean by DNA protein co-condensations? So, like this is a very well-known fact that when we keep on increasing the concentration of the protein molecules, we are actually increasing the interaction parameter, and uh, that results in the liquid phase separation. That's where these red proteins actually forms a liquid droplets, and that's what we call liquid phase separations. Now, on there is a different scenario where you can also tune where you can increase the interaction parameter by introducing some polymer to the system, or like polymer. By mean polymer, you can think of DNA or RNA, where this molecules can bind to this DNA or RNA, and as a result, what you are doing is you are tuning the interaction parameters, and as a result, it can form a similar droplet-like scenario, which in general what is called biomolecular condensates. And there are different factors behind these things, and one can give examples of like pure liquid phase separations to something of this sort of biomolecular condensates where DNA or proteins are also involved. Now this is what I call a co-condensation phenomena, because for this you need proteins which can self interact, and also you need a substrate or a DNA that can also bind to these proteins. And this is a condensation phenomena, but it needs a DNA or RNA to it. That's why we call co-condensations. Now in my talk I will stay in this regime where DNA and protein interacts to form these biomolecular condensates. Now, most of the co-condensation work that we see in literature is they are mostly concentrated, like related to the inside of the nucleus, where like the different proteins can loop the DNA. For example, it can loops the enhancer region to initiate transcriptions, and also other proteins like HP1 can compact or decompact the chromatin inside that of the nucleus. Now, in today's talk, I'm not going to talk about co-condensation inside that of the nucleus, but rather I will concentrate on the nuclear periphery. Because in the nuclear periphery also, like inside of the membrane, you have the chromatin surface, and then you have different nuclear inner nuclear membranes which can bind to the chromatins. And so you have this DNA binding proteins, membrane proteins, and membranes in addition to the DNA. So the question that one can ask is how these different DNA and other proteins and also membranes can in self interact themselves, and how do they organize in terms of the co-condensation, or in other words, like to be specific, like can DNA protein condensation on the nuclear envelope protect and shape the nucleus? And to answer this, I am going to use a single molecular approach where we have used optical tweezer, and I will show that using optical tweezer one can show different mesoscale effect that can ultimately lead to some organelle scale level phenomena that shows that indeed DNA protein interaction can protect and shape the nucleus. So before I go forward, I want to spend this I want to explain how uh, different proteins are organized on the nuclear envelope. So if you really zoom in here, what we have is the DNA, which is shown in the yellow color. And now the chromatins are bound to a protein called BAF. Now, BAF is a dimeric protein which can binds DNA to both of its sides. So as a result, it can cross bridge DNA. And then comes there is this magenta protein which is called LEM2, and this binds on the BAF. And this binds to the BAF on the one side, and on the other side, it binds to the inner nuclear envelope, membrane. So in a way, you can see these cascading structures, how chromatins are actually anchored to the inner nuclear membrane. Now, this, if you image the whole nucleus, this is also seen that these LEM proteins, which are only concentrated around the nuclear envelope, and the BAF is like they are enriched also along with the LEM2. Now, the another interesting feature of this thing is like the LEM2 can undergo liquid-liquid phase separations in vitro. So now the fact that LEM2 can binds to DNA through BAF almost provoked us to ask this question: can this combination also undergo some form of a co-condensation phenomena with DNA? So, like, is BAF DNA LEM2 co-condensation a driving force behind the chromatin organization at the nuclear periphery? Now, to answer this, I will use optical tweezer. A like just a brief introduction to the optical tweezer is like you can actually, so it's a highly focused laser beam where you can trap polystyrene particles. Now, in our experiment, we can trap two polystyrene particles which are functionalized with streptavidin, and then what we can do, we can catch a single piece of DNA in between two traps, and then what one can do is to stretch or relax these DNA particles and observe the binding of the proteins, because we work in a flow chamber where we can flush different proteins in different combinations, and we can also image the system simultaneously. So really we can observe the binding kinetics, and also we can measure the force and the force that the proteins apply on this DNA. So first we wanted to really establish this binding assay protocol and wanted to check how these proteins actually bind to the DNA. So the first thing that we added was BAF, and now you can see from this like the BAF actually binds to DNA, and you can see this cyan, the whole DNA is getting coated with these BAF molecules. And then we added LEM2 proteins, just LEM2 proteins, and you can add, as I explained, the LEM2 on itself cannot bind to DNA, and we don't see any signal on the DNA. But when we add both BAF and LEM2, and then we can see this, like this is where like you can see actually the LEM2 is chemically labeled here, but BAF is also present in this case. Now here both BAF and LEM2 is binding, and that's what we are seeing here. So this actually sets the binding assay that we are going to use in throughout the experiment, and we can see that the LEM2 actually binds to DNA in a BAF dependent manner. So the second experiment that I'm going to do is like we catch a single piece of DNA, and we will give a slack to the system, we will relax the system in presence of different proteins, and we want to observe how the system evolves. Now, before I go forward, I want to tell you how the force distance profile looks in case of naked DNA. So what you are seeing is like two beads which have a DNA in between these two beads, and what I'm doing is like I'm stretching and relaxing this DNA, and I'm observing how the force and the distance changes. So you can see here like the distance between the two beads is very small, the force is low, which means the polymer is completely slacked and it is mainly dominated by the entropic force. And if we keep on increasing the distance, what we are doing is like we are stretching the naked DNA just like an elastic rubber band, and it goes into this enthalpic region. And this whole behavior can be modeled as WLC, like warm light chain model, which is shown by this black dashed line. So this is a behavior of a pure naked DNA. Now, when we relax the DNA, we are nothing happens. Like I mean, we just see a slack in the DNA, and that is being reflected as a loss of signal in the DNA, because the DNA is actually going out of focus from the confocal imaging, and the DNA here is labeled with a dye through which we can see the DNA. Now, interesting thing happens when we do the same experiment with BAF in the first case, just BAF, and the second case is BAF and LEM2. So, what you are seeing is a co-condensation phenomena where you can see these droplets forming, but it happens only when DNA is relaxed. Otherwise, we don't see this droplet formation. And this is also reflect this droplet formation is accompanied by this plateauing of the force, where when we relax the DNA, instead of going into completely zero, the force just saturates at some force value. And this is because the extra slack that we are giving in the system actually goes into the co-condensate formation, and the system always maintain this minimum value. And depending on the protein combinations, this saturation force actually uh, mm, differs, and we can also find out the co-condensation energy from this. But then what we did is, like, we imaged the cell nucleus, and what we can see is there is an enrichment of the DNA on the nuclear periphery. And so what we think is, like, what we are seeing this as enrichment is actually the DNA co-condensation that is happening on the nuclear periphery because of these different proteins. So as I said, like, we can also measure the interaction energy, co-condensation energy from this different from this FD curve. Is like you can just integrate the area under this curve and calculate what is the energy that is stored in these co-condensates. Now, in one extreme, you have this just BAF, where the BAF actually loops this DNA and form these co-condensates, and this is happening because of the cross bridging property of the BAF. But when you have LEM2, this is the other extreme. You have the LEM2 binding to the BAF, and this LEM2 has this IDR which can interact, and that results in an extra force, and that is also reflected in the energy calculations. Now, in between these two regimes, there exists another regime where we have mutated the IDR region, like the intrinsically disordered region of the LEM2, where LEM2 just that in that mutant it can just bind to BAF, but it cannot self interact. So as a result, the energy is between BAF and LEM2. So in a way, you can measure this energy, and you can find what's the mm, role of DNA binding domain, and also you can differentiate the role of protein-protein interactions from these different kinds of measurements. So the third experiment that I'm going to do is to see how stable are these condensates. In this experiment, what I'm going to do, I am going to take a preformed condensates, and I'm going to stretch it, and I'm going to stretch it to unravel the DNA from the condensates. Again, I will just explain what happens when we stretch a single piece of DNA. So a single piece of DNA follows this worm-like chain model. Now, when you keep on stretching at a force of around 65 piconewton, the force plateaus out. That means, like, you cannot go beyond 65 piconewton, and what happens there is actually DNA, a nick is formed and a DNA starts to melt. The extra force that you are applying goes into the melting of the hydrogen bonds, and what we are seeing is a single stranded DNA as the dark region, which we are not seeing because nothing binds. Like, there is like, we haven't put any dye that binds to single stranded DNA. That's why this region is dark, and this is getting flanked by two double stranded region. So this is the main signature that the DNA melts at the 65 piconewton if we have some nicks. Now, this is the force profile when we stretch the preformed condensates in case of BAF, which is the cyan curve, and in case of magenta, which is BAF plus LEM2. Now, what you can see, if we keep on stretching, the sine curve actually marches the like worm-like chain model at around 40 piconewton. What it means, is like we can completely unravel the DNA from the BAF condensates at around 40 piconewton. But however, this magenta curve doesn't merge with this WLC line. What it means, like around this force of like 60 or 65 piconewton, we still have condensates that is not getting dissolved away. So then
There is a question. Pranay, can you hear us?
yes.
There is a question. Yeah, so the DNA intercalating dye that you're using,
Yes.
does it have any trouble in existing within the condensate in the DNA that's wrapped up by the by BAF and M2?
No, so I will come back to this question again. But what we have seen is like it can actually go inside the condensates.
So my question is there a difference in the quantum yield of fluorescence? Because for
Mhm.
intercalation, you need a little bit of B form DNA too, right? But in the
Uh,
condensate, it might not be in B form.
Sorry, I can't hear you properly.
In the condensate, all of the DNA may not be in B form, and that might affect the nature of the intercalation of the dye, and so the quantum yield of fluorescence of the dye. And so when you're seeing the fluorescence along the chain, just like you saw the dark patch for the single-stranded region where there was no dye,
Mhm.
you might see a lower intensity patch for the region where there is a condensate form, co-condensate form.
Exactly. So the DNA may not exist in B form, but what we are using here, we are not using any dye to quantify the amount of DNA wrapped inside the co-condensate. We are just using like force distance profile, and we can fit a worm-like chain model at different distance to find out how what is the amount of condensates inside the DNA, inside the co-condensates. But we are not using fluorescence profile to find out the amount of DNA, right? I mean, in this context.
You're doing some imaging, fluorescence imaging, right?
Yes, we are, but in this experiment we are not using that.
I have a question actually.
Yeah.
At that 65 piconewton, it is also known that maybe there's some models which suggest BDNA can also go to SDN, right?
Yes.
So,
So it depends on the DNA.
yeah.
So it depends on the DNA construct. If we have a nick, it always melts at 65
You have a
piconewton.
name. Okay, you have a name.
Yes, yes, yes. But it can also form sDNA, like which is like or sDNA at, but it happens also at 65 piconewton.
Yeah, please go
Hello.
ahead.
Oh, okay. Okay. So what it says is like the condensers are still are intact even if we cross 65 piconewton. And that means, like, I mean, from this force distance profile, we can actually quantify how much DNA is wrapped inside this co-condensers, and we can see that at 65 piconewton also, like almost 80% of the 75 to 80% of the DNA is still inside the co-condenser. What which means that the it's very difficult to unravel the DNA from the co-condensers. And one can also calculate the energies that it takes to unravel the DNA, and it shows that to unravel one base pair of DNA from the BAF-LEM2, it takes about 480 of work. So this experiment shows that the DNA is actually protected inside the BAF-LEM2 condensers. Now, this experiment actually also helped us to form this hypothesis is like the DNA inside the co-condensers are prevented against melting. Like we wanted to understand how the co-condensate is actually like what is the molecular scale that actually is helping to preserve these co-condensers and prevent it from getting melted. And what we can see here in this schematic is like when you are pulling the DNA from the both the end, the force actually gets redistributed inside through this protein network, and as a result the DNA inside is less stressed as compared to the DNA which is outside. What it means, like we wanted to check this, and this is we checked it using cytox as an intercalating dye, and this cytox is also spore sensor. So what we have seen is or other people have seen is this dye is recruited more on the DNA when the DNA is more stressed. So what you can see here, this is the yellow is the signal from the cytox, and what you can see is there is a high intensity of cytox, although cytox is present inside the co-condensate, as you can see from the yellow signal, but you have high intensity of cytox outside this co-condensate, outside these magenta signals, and what it that shows that the DNA outside the co-condensates are stressed more. And then we wanted to check that if the melting is happening, it should happen outside the co-condensates. And this we checked by a protein called FAS. And this FAS actually is a single stranded DNA binding protein, and that can only bind to us if there is any melting. And you can see here in this chymograph or in this snapshot that when there is a LEM2 condensate, the first signal is always binds away from the LEM2 condensates. And that says that the when we keep on stretching, if the melting happens, it happens outside the co-condensates. And that establishes our claim that the these co-condensates protect the DNA against external stress. But then the question is, like, how does this translates to the organelle level? And to answer this, we did a cell biology experiment where we compress the cell to create a scenario where the chromatin is actually under tension, just like the scenario in the optical tweezer.
Pranav,
And
there is
in
a
this,
question.
yeah, I will wrap up now. Then what we
Yes, you have a, but there's a question.
There is a question. I have
oh, sorry,
yeah.
yeah,
Can you go to the previous slide?
yeah, this one.
Yeah, the next one. No. The next, just the previous
Mhm.
slide. The next one. Yes. The next.
Yeah, this one.
Yes. So FUS, when you are saying that it doesn't go inside the condensate, it also depends on how FUS interacts with the proteins and so on, right? Maybe left,
Yes.
depending on the interaction energy, maybe it's just excluding them, right? That's a possibility.
Yes, so what we have checked in, I can show you some other slide, but what I can say is, like FUS, we have done an in vitro experiment where we have formed a LEM2 droplet and we have added FUS to it. And what we can see is LEM2 and FUS can coexist together
So that
in
okay,
the absence of DNA.
okay, thank you.
So I have a question to Pranav.
Yes,
What exactly are you calling melting? Because classically, melting is the breaking of the hydrogen bonds to separate the strands, which is aided by the fact that you have a nick. But which
yes.
part of it are you calling melting? Because from the diagram, it's not evident.
Yeah, so good questions, and this is also like we it puzzled us for a long time. And what we are saying is melting is it can be peeling of the DNA, which is because of the nick, and that peeling is actually like we don't have any very direct proof, but what we can say is like we can see different regions, different intensities of force. Like here you can see on this edge there is a high intensity of force, whereas in this region there is low intensity of force. Now this high intensity is actually because of the peeling of the DNA that happens due to a nick. And what we think is this low intensity region is actually a sDNA formation where the two strands actually separates out, where the DNA is intact, but you have only single stranded DNA which are in parallel.
You are pulling from both sides.
Yes,
You know that supercoiling of DNA can exist either as writhe or as twist. And
yes,
so when you stretch DNA, the twist changes essentially. Instead of being 10.5 base pairs per term, it changes, right? To become less.
yeah,
So when you have a nick, then the single stranded region allows complete rotation of the entire DNA molecule on both sides. So
yes,
when you are exerting force pulling away, then what you will have in that single stranded region is rotation. But it's
yes.
not very clear to me whether that rotation should also lead to breakage of hydrogen bonds. which is
So you are
melting,
right.
which is what melting is classically associated with. So what is causing the peeling is not evident unless the change in twist that is affected by the pudding is somehow also causing a melting.
Yes, you are right, and that also we observe. We cannot definitely say what forms of DNA, the complete, like what are the different forms of DNA at different regions in this melting region. But what I am claiming is like this fuzz accumulation on the DNA can only happen because of a single stranded DNA formation. We have we have control experiment where we have seen that the fuzz cannot have bind to B form of DNA at this condition. So whenever fuzz is binding, it is some other form of DNA other than B DNA.
Say that your data is more interesting than you currently suspect.
Yes, I
If you
mean
put your data to
we know that.
biochemistry physics, you'll find that this is indicating much more than people can. So the change in twist is actually also breaking hydrogen bonds. Okay. But Pranay, yeah, I'm sorry, partly I'm responsible, but
Yeah, yeah. I just
out
have
of
couple
time.
of slides more, and I will wrap up. Yeah. So what I was saying is like we compress the cell and we did experiment on two types of cell. One is in control and what in the other type of cell where we have silenced LEM2. And what we can see is like we observe high DNA damage when the LEM2 is silenced, and that we think is because of this LEM2, and that happens because of the co-condensation thing. The third slide that the next slide that I'm going to show you is when what happens to the force when this LEM2 binds. Sorry, when this BAF and LEM2 protein binds. And what we have seen is like in when BAF binds, the force doesn't increase. Like we held the hold the DNA at some 40 piconewton of force, and when BAF binds, nothing happens. But when BAF and LEM2 together binds, there is an increase in the force, and that increment in the force actually depends how stressed you are. Like more the initial tension, more is this increment in the force, and that tells me that like it is behaving some form of a springs. And now we went on to model this whole system as a springs in parallel or series, like where this DNA is a series spring which is with stiffness k1, and baflem to act come from the side and acts as a spring on parallel. And we can what it tells us is it effectively increases the stiffness of the DNA, and that is being reflected in this model. And that depends on this occupation fraction, which depends on the concentration. We went on to measure the stiffness of the DNA in presence of LEM2, and it matches very well with our model that you can see in this figure. So that means that this proteins acts as a springs. And but what does it mean in the organelle scale? Now to this is my last slide. So what we wanted to see is how these proteins are organized on the nuclear periphery. And you can see this it is like it forms a complete meshwork with other proteins that resides on the nuclear envelope. So we went on to model this nuclear shell, and we calculated the free energy of this. And this we did by two components. One is the bending and the stiffness component from any other proteins other than the LEM2, and this is the DNA stiffening part that is coming the contribution from the binding of these proteins. And then one can calculate the free energy. And what our model suggests is like when you silence the BAF and LEM2, the nuclear shape changes and it doesn't remain round. And this is also reflected in like obtained cell microscopy, and you can see that the nucleus is very buckled up when BAF is silenced in this case. So I will conclude my talk here. The different single molecular approach that we showed in optical tweezer led us to formation of these co-condensates, and that can explain why we have less DNA damage inside the cells. And also we can model these interactions with the springs in parallel, and that actually controls the nuclear morphology. Uh, and at last, I want to acknowledge my PI. Like I'm a postdoc in Stephen Grill's lab, and I'm also working with Alex, who started actually this project. And for the theoretical modeling, we are working with Frank. And I also thank Ramesh and Yahoo, who are the collaborators in this work. So thanks.
Thank you, very much. Thank you to all the speakers for sticking pretty much the time. Now we just have questions.
Okay. So Pranay, I'm just regarding the model. I mean, the first model is quite nice. It's a cute model, but I mean, the second one where you actually look at the, when you are just you have the Hellfish free energy and you couple it with this kind of spring model. So here, do you know all the parameters? That's one question. How do you extract them? Of course, it seems like you're playing with the different parameters and you're seeing what kind of behavior you can see, but can you be more quantitative in this? Because currently, the way it is, I don't know if you have really quantified the shape of the cells and or the nucleus, and then how the morphology is changing. And can one do this? Like say that I change BAF or LEM concentration in the cell somehow, right? I do
Yes.
RNAi or something.
So
And
currently, that's what we are doing. And what we have quantified the amount of BAF and LEM2 inside the cell. We have also quantified the area change, like we can quantify the like surface area of this each nucleus, and we can also quantify the effective spring constant that we get from the tweezer measurement, and we also have some other AFM measurement to actually quantify these different parameters that you can see in my screens. And currently we are actually investigating that part, and really we are actually like trying to put the values to each of these parameters.
Normally, the shape of something is a sphere. When the surface tension all over the sphere is uniform, right? And what might be happening here with the knockdown, I think you said, isn't it? Knockdown
Yes.
of BAF and then two
Yes.
is that the whole thing is becoming non-uniform and not behaving as one unit?
Yes. So
Spherical, sphere meaning, yeah.
so the knockdown of this BAF is actually breaking this cross bridging effect, and that is actually reducing the effective surface tension of this nuclear shell, and that is actually you are correct. Like what we are seeing is an decrease of the effective surface tension, you can say, but we are modeling it as an like decrease in the effective tension.
Question from Ranjit and also from Biman.
Hi, Pranay, very nice talk. I have a question. So you said the 60 piconewton force is that what's required to even partially disassemble the structure. And in cells, this would mean that the huge energy. And typically, is it? Are we aware of any mechanism to disassemble this? Because the cell has to not only assemble it, it also has to disassemble it, right? So
Yeah.
Are we know some ATP dependent mechanism that can disassemble that kind of energy driven process?
So what I can tell is there are measurements during mitosis where the microtubule are pulling that chromatin apart, and that pulling is actually creating some force of 100 piconewton of force. That is some cellular process, and that is beyond 65 piconewton first, and then when cells migrated through different constricted regions, there are measurements where that the force that is getting applied on the nucleus is also crossing that limit, and actually cell migration through very constricted region can actually cause DNA damage, that and there are other literatures to support this idea.
Question.
Okay, so Samantha had the same question.
Yes, I have the same question, that is it an active condensate from where the energy is coming? And my second question was, since there are multiple components in the condensate, is there any scaffold client type of interactions, like some components are very important and acting as scaffold and it can recruit clients. So do you think that fits in the scaffold client type of model?
So regarding your second questions, like it is actually uh, you are correct, and I can see. So if you see this slide, so here these co-condensates are not actually forming due to individual protein interactions. The first protein that interacts is the BAF that cross bridges the DNA, that provides the platform for these co-condensates, because LEM2 on its own cannot bind. And then comes LEM2. And so it's a like a hierarchical order of interactions that happens, and we don't know how the internal structure of the DNA inside these co-condensates, and that will be really interesting to check. And that's your first question was if it is an active, so it's there is no ATP or something like that in this co-condenser. So the whole co-condenser is a passive in some sense.
Thank you.
Thank you. Hemant?
Yeah, so Pranay, nice talk. I just wanted to know something, that the DNA you are using, is
Hmm?
that a specifically designed one? Because BAF is specific binder or non-specific binder?
So yeah, good question, and we have checked that. So BAF is actually is present everywhere on the nuclear periphery. That actually compacts the chromatin, and that tells you inherently that there should not be any sequence dependency. I mean, in a broader sense, and that we have checked in using two or three different construct of the DNA. The all the result that I showed is with lambda phase DNA, but we have also designed DNA where we have widum sequence in between that binds nucleosome, and we have seen like uniform coating all over the DNA always, and that tells me that like probably BAF is not a sequence dependent binding factor.
So Pranay, one last thing is that LAM, right? The LEM. LEM2, that binding is specific?
So the
BAF.
LEM2 protein directly cannot bind to DNA. It only binds to DNA through BAF.
Yeah, yeah. But with BAF, it binds to a specific pocket.
Uh, so the BAF binds to the minor groove of the DNA, and what we have also quantified that like under the stretch condition,
No,
BAF binds
So
every six
Pranay,
base pair of the DNA.
Pranay, he's asking if where does LEM2 bind on BAF? Is there a particular location on BAF, like position?
Oh, okay. Yes, there is a LEM binding domain which actually binds to the dimeric BAF.
Okay. Now, what I'm
Uh,
asking, you are
mhm.
showing also experiment and also imaging data, but maybe working with a buff which has weaker binding to LAM or something can directly tell you that this is what exactly is happening, right?
Yes.
Point, right? LAM and buff interactions. I know you have other experiment to show that this is what is happening, but let's say in my system I have buff which can interact with DNA but not with LAM2.
Mhm.
And I do the same experiment, then the force profile should not change with the addition of length.
I think what he's saying is,
Yes, yes. So in a way we have done this experiment, and like this force increase is actually due to we have other experiments to support this. Like this force increase that we see is actually due to lem interaction through IDR. So we have a mutant which can bind to just BAF, but we don't see this force increase.
Okay, okay, thank you.
yeah, very interesting, Pranay. Case of mutual crowding of BAF by DNA and DNA by BAF. Essentially, initially consolidating
Yes.
some kind of a condensation and then a hierarchical
Then
second
a
step
interaction.
involving LEM. Yeah.
That's like more like bridging. You're
Yeah.
creating a platform and then LEM comes.
LEM comes and make your elasticity and other stuff, the interaction stronger.
Yes, yes.
So thank you. We thank all the three speakers.